Afternoon to everyone still in other time zones. I hope you're still having a good morning. My name is Patrick Anderson, and today we are going to be talking about some bacterial diseases that affect trees. But before we get too far ahead, just some quick housekeeping notes. This webinar is worth 1 ISA CEU. So if you did not enter your ISA certification number when you registered for this, over there on the right-hand side of your screen, there's a little tiny box. If you click on that arrow and you go down there, you see there's something here that says questions. If you just type your ISA CEU certification number right in that box, we will record that for you and we'll make sure that you get CEUs. Likewise, anytime during the webinar, if you have a question, just type that question in that questions box and we'll have some time here at the end of our discussion to go over those questions, and we'll get them answered as best as we possibly can. And this webinar is being recorded, and you can find this as well as other of our previous webinars on our website under education. And we do have quite a few webinars coming up this spring that I'd encourage you to go on our website, treecarescience.com, and check those out. So again, my name is Patrick Anderson. I'm an arborologist with Rainbow Tree Care Scientific Advancements. So my job is really training and protocol support. So I travel around and I work with our Rainbow clients on getting predictable results utilizing our protocols around tree and shrub care. And anytime you can feel free to get a hold of me. You can also call our tech support line at any time. That number right there. And right there you can get questions on, again, protocols, on application rates, appropriate products, as well as to order products and get pricing. So again, that's the information there. We are here to really, Rainbow's goal is to better our industry and to work and partner with arborists and landscape managers all over the country to get practical protocols and great results helping trees and shrubs. Again, just last year, we did over 95 research trials. And we partner with a whole host of both private and public entities to, again, just provide the practical protocols so that we can save trees and shrubs in our landscapes and bring that to you. So today we're going to talk about bacterial diseases of trees, basically. And so we're going to review how bacteria affects plants as a pathogen. We're going to understand the life cycle of Fireblight. We're going to identify realistic protocols for management of Fireblight in the landscape. We're also going to understand the life cycle of bacterial leaf scorch as well as identify realistic protocols for management in the landscape. So to begin with, when we think about tree diseases and we're managing tree diseases, most of the time we're dealing with fungal diseases, and by far fungi are the #1 pathogen of trees and woody ornamentals. And if we go down the list, bacteria kind of falls as #4 on our list of pathogens. And about only 8% of tree pathogens are caused by bacteria. And there's some reasons for this. One is, when it comes to fungus and them being very good pathogens, they're #1 because, one, they're very effectively dispersed. So they come up with these structures so that they can spew their spores out into the world, and then they can find their plant hosts and affect them. And another thing there is those spores, they're armed with ways to actually penetrate into plant tissue. So they develop structures so that they can actually get into plant tissue and start damaging plants. And this is really why fungi are such a problem on trees and shrubs and plants in general, is because they're easily dispersed and they can actually physically break through plant parts and start causing damage. And this is counter to bacteria, because the reason why bacteria are #4 on the list is, for just about the reasons why fungi are so good, is the reason why bacteria are not great plant pathogens. And #1 comes, they're not effectively wind dispersed. So bacteria, they don't have structures, fruiting structures that allow them to get out into the atmosphere and find plants to affect. They require vectors, things like bees and leafhoppers, to get into the plant themselves. And then secondly, they don't have those structures to get inside a plant. So again, they need to find an opening. They can't just land on a leaf and start going to work on damaging that leaf. They need to find entryways into the plant. So things like plant wounds through pruning or hail, again, vectors, so insects feeding and damaging the plants, actual plant parts like the stomata and the hydrothodes, which are specialized parts of the plant to excrete water. So the bacteria actually needs to find a way inside the plant. It can't break through the plant in a matter of sense. And really, when we talk about bacterial diseases in our landscapes, most of the time we're talking about two different diseases here. We're talking about either bacterial leaf scorch, which we can see here on the right, or Fireblight, which we see here on the left. And that's what the rest of our discussion is really going to be around, the biology of these pathogens and practical protocols for managing these pathogens. So we'll start first with Fireblight. So Fireblight has a pretty broad host range. We often think about Fireblight on things like our pears, our ornamental apples, as well as edible apples, other stone fruits, cotoneaster, loquat for those of us down in the southern regions, and hawthorn, these types of plants. Fireblight is a worldwide phenomenon, and it's really, Fireblight is really a huge deal in orchard production, as I'm sure y'all can imagine, where it can cause over $100 million in damages alone. This is just in the United States. And again, this is a worldwide problem. If you think about the amount of orchids that are out there worldwide, it can be a real devastating pest. And so when we talk about Fireblight and how we begin infection, most of our infection begins in the flowers. So as I mentioned, with bacteria need to find a way into the plant. They can't break through the plant to start causing damage. So the flowers have these little things called nectarthodes, where nectar, of course, is secreted through, and this is a great place for bacteria to be introduced. So our own invasive species, the European honeybee, is actually a great vector for Fireblight, as they are very active pollinating flowers in the early spring on many of these species that are susceptible to this disease. So this is how a lot of our Fireblight infection is being spread, just by pollinators. And then we also, as far as the environmental factors, we need cool, wet weather, not unlike many other diseases that we're familiar with, like our fungal diseases, but we need cool weather, as well as this plant pathogen, and we'll see this here in a moment. It can be spread effectively by rain splash around a plant. So you can get an infection site on the plant, and then we get a rain event or an irrigation event, and then the pathogen can be spread throughout the plant that way. And as I mentioned too, plant wounding, so pruning and hail damage. Often in early spring in parts of the country we're susceptible to early spring hail storms, and this can damage plant parts and again cause an entryway for this pathogen here. And so just to kind of look at how this thing spreads, it moves in here, it colonizes a flower, and this is what you see these little kind of these rod-shaped structures here. These are our little bacteria, and very quickly, 48 hours later, this is what we have here. So this is why we can see rapid decline and damage in plant parts after they've been colonized by this bacteria. And outwardly, this is what we often see. So, our bacteria here goes through a canker phase that can girdle and kill whole parts of the plant. This here is usually our first thing that we often see is the shepherd's crooking. We're starting to see necrosis here in branch tips. We'll talk about this here in a little bit. This flower blight stage where flowers start falling off, and you can see here this kind of necrosis. And then also in the fruit, we can get this kind of bacterial exudate coming in on the fruit here. And these are all kind of the visual signs that as we're walking past the plant in the landscape, a client calls us, they say, hey, my crab apple isn't looking bad, or my pear isn't looking good. These are the signs we're often seeing to help us diagnose what Fireblight looks like in the landscape. So more in here onto how the life cycle of this disease is. It overwinters in these cankers. So these cankers are key to identify and isolate, because this is where the bacteria is going to overwinter. And then in the spring, as it starts to warm up, they begin to ooze this bacterial exudate here. And this is just full of bacteria that's just, aching to move other parts of the plant and other plants to start causing damage. And as I mentioned, this can be caused often by rain splash. So we can get a canker that looks like this, and we get a rain event or irrigation event. This bacterial ooze is then splashed in other parts of the plant, on the flowers, onto recently pruned or damaged areas of the plant, and then it can move in and start causing damage. This stuff's also kind of sticky, so again, we can move it, insects can move it. And this is the way bacterial diseases are really spread. It's really often by some kind of outside vector. And if we look at this really complicated chart, to the ways that these bacteria spread, this pathogen is spread, it starts here where we have an insect like a European honeybee, which is not native to the United States, being attracted to this bacterial ooze, and then of course it goes and it lands on a flower, and then the bacteria is allowed to enter into this flower. It starts to multiply very rapidly, causing damage to plant cells, and this is what we often see. We start seeing these kind of infected flowers begin to change color and shrivel, and then that moves down into the spurs and twigs, and then finally that moves down actually into the plant, and that's where we start getting these cankers to form, and of course we can start seeing whole branches die off because of this girdling, and then again the fungi overwinters in this canker and the process starts all over again. Another way that we often see Fireblight affect trees, again especially in the spring, is when it moves in on these twigs here. So this tender twig growth, this is fast expanding twig growth. It's yet to become woody and harden off, as we say sometimes. It's the soft new growth, again, which we have, and kind of spaces in between this new growth where this pathogen can affect. So again, imagine we have some kind of a rain event here. It splashes onto this tender twig tissue. It moves into these cellular areas here, and then again begins to multiply rapidly. And again, the infected cells begin to collapse. And once again, we have this canker formation, which again can cause girdling. Here, whole branch death, whole tree death in some occasions. And again, we get this canker. And again, it started all over again the next season. And that's the quick kind of view of a management, or the life cycle of bacteria. So what can we do about it? I'm sure we've all seen something that looks like this before. We're all probably familiar with this disease triangle. And basically, in this disease triangle, as we all are probably familiar, we need three things to have a disease. We need a susceptible host, like a crabapple. We need a conductive environment, like cool, wet weather. And then finally, we need a pathogen. And when we have these things, we have a disease. But, in any of our disease management strategies, if we can alter this in any way, then we're not going to get a disease. So, it's not always, we often, with our disease management especially, we often kind of default to some kind of chemical control, and we have great protocols using these products to control these plants, but if we think outside the box, we can do some neat stuff. So when we talk about changing the host can be difficult, but if we're also thinking about some of these species, especially some of these ornamental species, one crabapple in somebody's front yard, and as long as it wasn't planted by their great-great-grandmother, who once met Abe Lincoln, then what's the harm in maybe just removing that plant and putting in something that's going to be resistant? It's going to be a lot easier for your client, because again, you're going to go out there and say, hey, we're going to spray this plant 3 times a year for $100, or for like $500, we can solve your problem by putting on a whole new plant. It's some outside the box problem solving. But even there, we have in this case here, looks an example of apple trees. We have some apple trees when it comes to Fireblight that are moderately resistant, but you can see here we don't have any real apple trees in this case that are completely resistant to Fireblight. So if you have a plant that, again, from a genus level is susceptible to Fireblight, under heavy disease pressure, you will still likely come up with disease. That's important to know. Some other things that we can do about modifying the environment. This guy's kind of like modifying the environment, modifying the plant, but a big part of our management protocol, regardless, especially when it comes to Fireblight, is going to be removing those diseased parts of the plant. So pruning out those cankers, because if we remove that canker, we're removing a sink for the disease, and again, we're going to drastically reduce the amount of inoculum that's going to be out there. So removing the canker is a big part of our protocol, regardless of what strategy we decide to take when managing Fireblight. So we call this sanitation pruning. Now, again, I mentioned earlier, we need to be careful because we can actually, by pruning the tree, we can create new pathways for the pathogen to enter. Likewise, if we are pruning the tree during a part of the year where that bacteria is going to be active, we have the potential, albeit slight, but we have the potential to move that pathogen with our pruning tools to new sites and basically infecting the tree. Again, this is a rare occurrence, but certainly there's a potential there. So our best management practices or our protocol for pruning of Fireblight would be we want to do it in the dormant season when that pathogen isn't actively around to infect new sites. Secondly, we want to prune. We try to avoid pruning through the canker. So ideally, we would be below that canker to some extent. And again, I think this is arguable depending upon what you read and who you speak to, but anywhere from 6 to 8 to 12 inches below that canker, just to be doubly sure that we're not cutting through the canker and again, reducing our risk of spreading the pathogen. One thing that comes up often is, what about disinfecting my tools? And again, there's some research out there that talks about different ways of or different products and protocols for disinfecting tools. But this was a piece here that I just took from the Linda Chalker-Scott out there in Washington and her blog, Garden Professors. And so here are some options for disinfecting tools. You can see alcohol wipes, Clorox, so bleach, some of our household cleaner, Listerine I thought was real interesting, but Lysol spray, and Pine Sol. And then something I read not too long ago said, I don't know if it was necessarily a peer-reviewed trial, but anecdotally speaking, the Lysol spray was not as hard on the tools as using Clorox bleach. So the Lysol spray, again, you can buy it, spray it, is a good option there if you're worried about passing along pathogens in the environment. Another thing to keep in mind is that, especially we've seen over the years, that heavy nitrogen fertilization can increase disease occurrence. And part of that is because heavy nitrogen fertilization will increase the amount of tip growth that we're seeing. So we'll have an abundance of that kind of soft green twig growth. So, we do see that nitrogen fertilization can be beneficial for tree health in some cases. But if there's other ways we can make the plant healthy and try to avoid putting on so much of that top growth that then can be infected by the bacteria, that's a pretty good route to take. So simple things like, these cultural practices like correctly mulching our tree, and I know, we probably all know the great things about mulch. But again, just correctly mulching the plant, making it healthier, making it happier, not pushing all that new growth. Other things we can think about modifying the environment, again, going back to the fact that our bacteria, it needs either a vector or it needs to be rain splashed to new plants or new parts of the plant. So, it's hard to stop rainstorms from happening. I don't think there's a lot of us going out and tarping all of our trees during a rainstorm because we don't want to spread fire blight. But things like this, easy things like fixing our irrigation. So we had overhead irrigation, we had cankers on a tree, we can splash that bacteria all around all different plants. So if we could just alter, again, our environment, alter our irrigation practices, then again we're going to reduce the incidence around the tree. And then finally, of course, we always go to this idea of, well, our chemical control, our foliar control. And we have a lot of great options for foliar control. The key to this, of course, is the devil's always in the details when it comes to applications of any product. And of course, this here, timing is going to be so crucial when doing these types of foliar applications, spray applications to manage fire blight. And so, we have a couple of different options. One is we have antibiotic sprays. Streptomycin is a real popular one used. And so what we want to do here is we need to spray around the time the pest is active and during events where the pathogen can actually make it into the tree. So what did we talk about? One of the best ways that the pathogen makes it into the tree is through bloom. So we need to definitely treat our trees around blooms. So spraying around blooms, and this streptomycin would work. It has a prophylactic type function there. So it's protecting the bloom from infection from bacteria. Other times to spray, again, after some kind of event that would wound the tree, hailstorms or windstorms. We have a lot of storm damage due to some kind of event. We would need to spray then if, again, the temperature and the weather are conducive to the Fireblight management, or excuse me, the Fireblight infection. So that's another time we want to talk about that. And again, it's this environmental conditions that favor disease that are so key to when that pathogen is going to be out there. So again, that cool, wet weather, we're going to have a lot of pathogen development. And again, keying in our sprays around the time the plant is blooming. And then if we have some kind of event where we're getting damage to the plant, making sure that we're getting those antibiotics on the plant parts that are going to be affected to block that disease from ever entering the plant. Now, of course, we all have heard some issues with antibiotics. Even in the management of us human beings, we know that we can actually, some pathogens can become resistant to antibiotics. And so basically what we have found here is that the same is true of diseases. And so we definitely have a potential, when overused, to develop a resistance to some of these antibiotics. And you can see here, streptomycin is by far probably the best antibiotic to use, but in some cases resistance has begun to develop. And again, it's because it's used so much throughout the world. And really, this is, I have to preface, this slide is really, it comes from agriculture, orchard production. It probably would be a bit of a stretch to get a disease resistance in the landscape. Certainly a potential, and something we definitely need to think about. When we're managing our landscapes, to be responsible landscape managers. So another foliar applied chemical control, if you will, for Fireblight would be these copper sprays, which again, many of us are probably familiar with copper sprays. Copper can be used for many different pathogens, both fungal and bacterial. And so this has been around for a long time. People have realized the benefits of copper. I mean, probably going back to ancient times. We call this the Bordeaux mixture, has been called in the past. And because of the way it works on the pathogen, little resistance has developed from this. So this would be this copper spray regimen mixed in with your antibiotic sprays. Isn't a bad idea. Again, we always talk about rotating pesticides, and in this case here, these are two tools that work very well when applied, again, at the right time of year and at the right physiological moment in the plant's development to really hold off these diseases. And so, again, our protocol here for this copper spray management is we're targeting this kind of this phase here, this flowering phase. So, our standard protocol starts with a what we call a dormant treatment. So we are mixing at a very high rate of copper, and we're mixing with the horticultural oil. And the horticultural oil does two things. One is it works as a surfactant to help the copper penetrate these bark furrows and these cankers to help to kind of combat the disease in that way. As well as we all know, horticultural oil works very well on insects. So we kind of get two bonuses there. We're both helping, the oil is both helping us to stop the infection for the next season's growth, as well as taking out some plant-damaging arthropod pests as well. And then this would be followed up by a second spray of copper at a lesser rate, and this could still be mixed with oil or some other surfactant. I always recommend with any of your foliar applications outside of oil, always put in some kind of surfactant, because it's just going to improve the quality of your spray. And so our second time that we're spraying would be, again, right here at peak bloom, again with the knowledge that we're going to get a lot of this disease vectored by these pollinators, and what we're doing is preventing this flower blight phase here. And this is again, if you can get your timing on this right, it's very, very effective and a great way to manage fire blight. Also known that fire blight can be tricky, and we have some years where we have very, very high disease incidence. And again, it can be something that's very damaging. It looks very bad. Another way to manage this through chemical controls, if you can't spray for whatever reason, and we can certainly all think of situations where spraying is just not necessarily the greatest option, we can actually systemically inject an antibiotic into the lower trunk of the tree, and it will be uptaking the tree, and it will provide some protection against the plant. And again, this is something that needs to be keyed in on timing. So just after full leaf expansion, or I even say just before full leaf expansion, get this product injected in the tree. And again, we want to get in there before symptoms are present. And this is, again, this is oxytetracycline. This is our BACASTAT. And the way this is working is it's basically, we talked about how we get some infection here on these twigs. So we have these expanding green twig tissue, which can be very susceptible to bacterial, or excuse me, Fireblight. This product moves into this area here and stops the bacteria from colonizing these intercellular areas, which then leads to cell collapse. So what we will see, our expectations when we do an injection of BACASTAT is we will still get this flower blight, but what we are preventing is this infection moving down to the flowers, into the spurs, and into the twigs. So although we might still see these kind of, again, this flower blight, these kind of brown, dark flowers that are held on to the tree, the pathogen is going to be prevented from moving down into the plant, where, again, we're going to get this canker formation, which, of course, can then girdle whole parts of the tree and twigs. So this is very, very effective in this. And we can see here, this was taken from a trial where we have oxytetracycline, which again would be our BACASTAT, and our water-injected control. So we have oxytetracycline. We can see the amount of shoot blight severity. And so we're getting about 10 to 15% of our canopy affected by shoot blight, versus with just our water injections, we're getting there between 60 and 70%. Likewise here in the next year's study, you can see there was a lot more disease activity that year, but still we are drastically reducing the amount of tip blight with one properly timed injection of oxytetracycline, which again is our BACASTAT product there. Another thing that we see, of course, going back to the idea of modifying the host, if we can apply a growth regulator like Cambistat, we will induce some things within the plant to help it be more resistant against disease. And especially here, we find, and there's research here by Dr. Herms that shows that if you can apply something like Cambistat, a growth regulator, it'll actually, while it is reducing the amount of vegetative shoot growth, so it's reducing the hormone responsible for top growth, we see an uptick in the defense compounds. And again, this was something shown in several papers where we're seeing an increase of defense compounds when we apply a product like a growth regulator like Cambistat. And so another thing that we're doing here is, one, we're seeing an increase in defense compounds, so the plant is able to defend itself a little better against pathogens. The other thing that we're doing when we apply growth regulator is we're basically, with this fast-growing green growth, we're getting a lot of entry points for bacteria, versus if we have a lot less growth and a lot less opportunities for this bacteria to get into. So, physiologically speaking, we're increasing defense compounds. Morphologically speaking, we're creating a less conductive plant, for the bacteria to even invade. So this is a really neat option here. And here's some trials. This was done with Dr. Frederick at the Bartlett Research Labs here in Charlotte. And so we can see here, our blue is our control treatments, our red is the coside, which is copper, and the yellow is our foliar applied paclobutrazol, and this is the blighted shoots. So again, we can see that our copper treatments hold up are probably the best, but in comparison to a foliar application of a growth regulator, we are still getting great control compared to our untreated trees. So another great option, another tool in the toolbox, and again, something that can be combined with our other protocols to really prevent fire blight. Here we have another trial here. This is in 2008. And so again, in this trial here, we used, the Bartlett research folks used a lower rate of copper to see if it would have any control. You see that low rate of copper actually had more disease shoots than our untreated controls. So definitely follow the higher label rates for your copper applications. But then again, compared to our Cambistat treatment in this case, we are getting significant difference in the amount of disease shoots versus our untreated control. So again, the use of Cambistat and Trimtech either as a soil application or a foliar application to reduce that tip elongation, to reduce the amount of sites that the pathogen can affect is a great option for us. And so this is what we have for our protocol options here. Of course we have our copper sprays. I apologize, I didn't put in our antibiotic. We have antibiotic sprays. We have our copper sprays. We have tree injection with our antibiotic with BACASTAT. And then, of course, we have our growth regulators, which, again, we've seen in all these cases can really do a great job of controlling the fire blight. Again, it all comes really down to timing and season of doing this. So now let's segue into bacterial leaf scorch, our other bacterial disease that we run into very often. So this here, Xylella fastidiosa. This pathogen they've actually found on probably over 100 different species of plant. So this bacterial leaf scorch, this Xylella, can be found in a lot of different plants. But for whatever reason, it really only turns up. We only see the damaging symptoms and the damage in a handful of plants, and namely trees in our red oak family, like our pin and our red oaks. We see it in sycamore, occasionally in elm, occasionally in some of our maples. But you can see here it affects a lot of different plants. But it really only, again, it seems to only damage a handful of species within certain genus, which is really interesting. But also plays to our management. It's tough disease to manage because of the way it affects plants and the way it can be found in a lot of different plants. So again, going through a quick kind of look at how this thing is spread. So bacterial leaf scorch is primarily spread by insect vectors. So most often these leaf hoppers, we call them sharpshooters, are the ones that are going to spread this disease. So what they're going to do is they can feed on a plant that has Xylella, and let's say, again, knowing that there's a lot of different species, this little leaf hopper could go feed on a plant that's showing no symptoms of bacterial leaf scorch, might never ever show a symptom of bacterial leaf scorch. It can pick up the bacteria, it can hop over to a pin oak or a red oak, it can start feeding, and as it's feeding, it's going to kind of basically, in so many words, kind of regurgitate some of its guts back into the plant, and that's where the bacteria is going to be introduced. And now that tree over time will start to display symptoms, and it can be very, again, a very dramatic appearance of this tree versus unaffected trees. It can be very damaging. Again, so this is what we were looking at here. This insect lands on that leaf, it starts feeding in that vascular bundle, it's feeding that xylem area, and of course this is where, again, the bacteria itself can't penetrate the plant, but with the help of this little leaf hopper, this little, again, these guys are kind of neat looking, this little tiny leaf hopper, that's a leaf hopper nymph. Here's I'm not sure the exact species of this guy, but he's really, look at that neat little green stripe on him there. Little tiny little guy is not doing too much damage to the tree, but here they're spreading this disease. And again, what we haven't mentioned yet is this disease, is bacterial leaf scorch. There's no cure for bacterial leaf scorch. Basically, once the plant has been infected by bacterial leaf scorch, it will always have bacterial leaf scorch, which is a real kind of bummer when we're managing some of, especially these large, beautiful old oak trees and some of these properties. It can be a real shame when we start noticing that, hey, this is what our problem is. It's BLS. And so basically what happens, again, very much like our Fireblight, the bacteria is introduced, it starts to produce very rapidly. And so what it's doing is it's blocking up intercellular spaces, so it's blocking water transport. The part of the reason too why some species are more sensitive to others is the way they react. So they actually, in our oak species specifically, there's an overproduction of these defense compounds, and so they put up things like tyloses to block the disease because, again, they realize they're infected, they're trying to stop this disease, but in stopping the disease, they're also blocking off water transport within the plant, and of course these embolisms form, and again, this is all plugging up xylem vessels, so again, they can't get water and nutrients up into the leaves, and this is where we start getting these scorch systems from. That's why, again, they call it bacterial leaf scorch because it looks like an environmental scorch. It looks like drought symptoms. But it's not drought, it's the fact that the poor plant is blocking water from getting it up into itself because it's trying to fend off this disease. Very interesting. And again, this is the way this disease can manifest itself. We have here, again, just different examples here of oaks. This is pictures that I borrowed from the Missouri Botanical Garden. Again, this looks like it might be a drought issue, maybe a root damage issue in this case here, but it's this bacterial leaf scorch, which is it's a shame. But again, if we start talking about protocols for this, again, there's no cure for bacterial leaf scorch. There's nothing where you can really spray to prevent this disease. So it really comes down to trying to suppress the spread of the bacteria in the plant and then suppress the symptoms of bacterial leaf scorch for as long as we can, because it is the early defoliation of the plant, because what we find is when a tree has been infected with bacterial leaf scorch, we start to see it defoliate early on in the season. So again, like in the mid-Atlantic states, we start seeing trees defoliate the end of July. In other parts of the country, they might even start defoliating as early as the end of June. And so we're losing photosynthetic area for a large portion of the season, for over half the season in some cases, which of course will eventually lead to, again, it begins that spiral decline in the plant, and that's really why this bacterial leaf scorch can be such a big deal. But again, so how can we manage it? We need to try to suppress those symptoms for as long as possible. One way to do that is, again, injecting an antibiotic directly into the tree. And again, our oxytetracycline or BACASTAT, directly injecting it right into the tree to suppress those symptoms for as long as possible. And so what we want to do, ideally, is we want to inject it after full leaf expansion, but before, July 1st is kind of like, again, our soft target there. Really get it in the plant before it's starting to show symptoms, try to suppress that disease. And here's a trial here. This was done with Dr. Hartman in the University of Kentucky. And so basically, as I said, we can't cure this disease. We are eventually going to get symptoms. But what this chart shows us here is that when we inject our BACASTAT or oxytetracycline, we can suppress those symptoms longer into the season. So we can see here, we're starting to see symptom development in these trees in August in our untreated controls. And then you can see the amount of, this here is the amount of canopy that is being affected. So we can see it's drastically going up in our untreated controls like our oxytetracycline here, where we are suppressing those diseases for a greater amount of time, or suppressing the symptoms for a greater amount of time. And so that is our goal there, is to suppress those symptoms. And if we see some pictures from this trial here, you can see this was a pin oak that was affected by 90% leaf scorch in 2006, and then it was injected in 2007. And so you can see here, a year later, the difference in the canopy here. So again, this plant still has BLS. It's still going to defoliate before its neighboring uninfected trees, but we're able to hold on to those leaves for a whole lot longer. Likewise, here's another example of a pin oak. Again, this is 75% affected by a leaf scorch. Here again, a year later, drastically reduced the amount of symptom development we were getting in that plant. So it's a great way to do it. Of course, we're talking about here just trying to maintain tree health, and that's one of the best things we can do. So outside of the injections of OTC, and one of the downsides of the injection is it needs to be done every year. You need to inject OTC into the plant every year, whether it be for Fireblight or for the bacterial leaf scorch. We're only getting one season of disease suppression and symptom suppression with these injections. So that's where, with BLS especially, it's just a matter of keeping the tree healthy. So doing things like proper prescription fertilization, making sure that our soil chemistry is right, making sure it has the adequate amount of nutrients in there, making sure it has the adequate amount of organic matter in there. Again, all the great benefits of mulching. Try to get that grass competition, other competition away from the plant, get mulch down there. Doing things like our root enhancement system, where we're taking our air spade, we're decompacting the soil around these trees. Again, just kind of changing the soil from a damaged soil, trying to mimic more of that forest soil that these trees have evolved to grow in. So using our air spade, putting in prescription organic matter and soil amendments, watering the plant, adding a growth regulator, and then finally correctly mulching and then watering the plant. This is really one of the best things you can do for stressed trees. And again, it's just going to help the plant to alleviate some of these, drought-like symptoms that are going to be induced because of the disease. Again, the incorporation of a growth manager, like here again, BACASTAT. So what we see at BACASTAT again is we're reducing the hormones responsible for cell elongation. We're increasing other hormones that can be very beneficial to the plant, as well as we're increasing chlorophyll production, because part of what's happening is we're taking elements away from growth, above ground growth, and putting them in other parts of the plant. So we see an increase in chlorophyll, which again is going to help us to alleviate these scorch systems for longer. And so I realize this is a busy chart, and we got a lot going on here, but basically what we can see is, with our untreated controls, we get symptoms starting earlier in the year, so again, starting on early August, and then getting gradually worse as time goes on. But with, again, we have our BACASTAT treatments, we have our BACASTAT alone treatments, and then we have our BACASTAT mixed with BACASTAT treatments. And what we're doing is we're holding off symptoms for a longer period of time, and generally speaking, they're not becoming as severe. And that's what we can do with bacterial leaf scorch, is just do our best to make the tree as healthy as we can for as long as we can, hold off those symptoms for as long as possible. So one thing that we used to talk about when I started, back in the early 2000s, one thing that we started doing was we tried to do vector management. So there was a theory out there that if we put imidacloprid in soil, then we would hopefully kill these leaf hoppers and these vectors before they could pass on the disease. But a lot of this stuff, this is called Pierce's disease in grapes and viticulture. So a lot of work has been done in viticulture, and basically what they have found is that there's no solid evidence to support this vector control theory. So the thought is by the time the insect begins feeding and is ingesting a systemic insecticide, it's probably already passed along the bacteria into the plant, which is unfortunate. So the consensus says basically it's not practical. One thing there is it might be used, you can maybe create a sink there where you have a tree that has bacterial leaf scorch, and basically what you'd be doing if you applied something like imidacloprid, you would be killing a bunch of vectors, so that would reduce the amount of vectors out there to spread the disease. So there's one thought there of why you would do that. But as far as protecting trees from being infected, that's probably not going to be a very practical way to do that. So, in review, we had Fireblight infects trees in several ways. Again, our bacterial diseases do not have a way to penetrate a plant surface. It's going to come down to some kind of a vector, whether it be a honeybee, whether it be a leaf hopper, or whether it be a pruning tool. This is how our diseases are going to be spread. We do have several predictable protocols to manage Fireblight. Bacterial leaf scorch affects many plant species, but again, it really only shows up in a handful, and unfortunately some of them are more coveted landscape species. The BLS is going to be vectored by one of these leaf hoppers, a polyphagous leaf hopper, which means they feed on multiple different species. So again, it can land on a plant that's not showing any symptoms and may never show any symptoms, and then spread it to a healthy plant that can show symptoms. There's no cure for BLS, and there's no real good preventive measures to prevent BLS from affecting your plant. So if your plant comes down with BLS, it just comes down to managing the symptoms and slowing the pathogen's development and other tree healthcare protocols. With that, that concludes our discussion. I will let you guys know that we have several other webinars coming up, so please visit our website at treecarescience.com. And you will see that we have, again, a whole host of really great protocols coming up. The other thing I failed to put in this presentation, and I apologize to everyone, is the saluting branches. If you're not familiar with saluting branches, we will be doing saluting branches again this year. And so saluting branches is a nationwide community event where arborists and landscape managers converge on Veterans Affairs cemeteries. A lot of these cemeteries don't have good funding, and they have a lot of work that needs to be done And so arborists and landscape managers converge on these veteran affairs cemeteries, and we donate a day of service. We did the first time last year. It was a great event. It was very rewarding. We're doing it again this year. It's going to be in September. You can go to salutingbranches.org, and you can learn a whole lot more. And so with that, I will go and take a look at our questions here. And if there aren't any questions, I, of course, will stay on here and hold on. Let's see. So our one question here is, crab apples will flower before leafing out, when to inject? And so, again, when we're injecting oxytetracycline BACASTAT, we are not going to be able to prevent that flower blight. So that's a key distinction to know. When we're injecting for Fireblight, we're not going to stop that flower blight phase of the Fireblight. So the flowers will still get blighted. You'll still see that kind of turning kind of, again, brown and holding on. But what it will prevent is it will prevent the bacteria from moving past those flowers into the spurs and into the twigs, and it will reduce that shepherd's crooking, and then also reduce the chance of getting canker formation. So you're right. Yes, the flowers will come out before the leaves, but what we're doing is we're trying to slow that spread into the spurs and branches. And so I will hold on here for just a few more moments if there's no other questions. I'm sure nobody would mind getting off the line early today, but we'll hang on here for another minute. So we have another question here. Is there a way to check the difference between BLS and sun scorch? So yeah, so basically what's going to come down to is you're probably going to need to get a lab analysis to really distinguish between BLS and some other type of scorching disease. Some things of note, you know, BLS is going to, when it starts taking hold, and actually maybe we can go back in the presentation and take a look at some of these leaf pictures. BLS is going to have a distinctive type of pattern when it affects the leaf. And so what we see is this here. So it's going to start out on these kind of leaf tips and move its way down kind of in between these veins. And that is very distinctive of bacterial leaf scorch. Some other things, and this isn't always prevalent, but in many instances, and if you get in the literature, they'll talk about having this yellow-looking halo around the leaf. I stopped presenting that only because I found that not to be true. We would get positive BLS analysis. We get our samples analyzed in the lab, and they'd come back as positive, and they didn't have that distinctive yellow halo. So I felt like I was doing a disservice to people by presenting that, because again, we found very often we didn't have that distinctive yellow halo. But this is going to be what bacterial leaf scorch is going to often look like. And so if you have a tree, again, if you have a tree that doesn't have any signs of root damage, you have a relatively normal year with you know, adequate moisture, and you start to see these symptoms, especially coming on kind of that early summer time frame, that kind of late June, July time frame. If you start seeing this in part of the plant, that's a good indication that you have BLS. And then what you're going to need to do for laboratory analysis, and this may differ based upon the lab you work with, but generally what you're going to want to do is collect 30 of these leaves, both a combination of 30 leaves, symptomatic and not showing symptoms, and collect them and include the petiole. So include the whole petiole, not just like some torn off pieces of leaf. And again, it's usually 30 leaves combination of showing symptoms and not showing symptoms with the patio, and then mail that off, and they'll do what they call an ELISA test to isolate that bacteria. And we'll give it another minute here to see if we have another question or so. All right, guys. I'm not seeing any other questions coming up, which is fine. Again, thank you so much for your attention. Again, please go to our website, treecarescience.com, and you can learn more of the webinars coming up. If you have any other questions, please feel free again to contact us at our tech support line, or you can always contact me directly. I really appreciate everyone's attention today. I hope everyone has a great afternoon, and we look forward to seeing you sometime in the future. Thanks a lot, everyone. Have a good one. Bye.